Hi everyone, welcome to the fourth episode of my tutorials on Nextflow for bioinformatics. In today's video, we are going to look at how to write a Nextflow script to perform genome assembly. All right. Now, before I proceed, let me say that the script for this tutorial, the materials for this tutorial are all available. So check the description box, you'll find the links to the materials and then you can download them. All right, so that's what you need to know. Now, this is what we are going to do in this tutorial. Let's just look at it. We are going to use the tool SPADES for the genome assembly. I mean, there are different genome assembly tools available. We'll use SPADES. And then for the mode, we'll use chromosome. Now with SPADES, you can assemble plasmids. You can also assemble chromosome. So we are going to use the chromosome mode. For data, we are going to use Illumina paired end reads. We will download them, by the way, so don't worry about it. And for sequence type, it's going to be whole genome sequence. So that's what you need to know. Now, for this tutorial, you need to make sure that you have Nextflow and SPADES installed. And I have tutorials that show how to install these two. So check the description box, you'll find the links to those videos. So these are the requirements you need to have. All right, now let's get started. So we are going to open our terminal and then start the exercise. Okay, so we are on the terminal now. So what we are going to do is to first create a working directory and cd into it. So I'll say make the Nextflow, and then I'll cd into it. All right, so I'm doing this because I don't want to mix up files that I generate from different projects. All right, so in this directory, if you have, let's say, multiple Nextflow projects, then you can create another directory called genome assembly and then work in it. But we will keep it simple, just like this. All right, so let's do this. Now we have created our directory, so we are ready. So what we are going to do next is to download the example data. We will do this in Nextflow, but we need to get the download links for the files. So let's go to the ENA page. So we'll visit this page here. I'll leave the link to this page in the description box. All right. Now, when you are here, come to this side. Okay, where we have this. You see this accession ID here. You may not find it there. It will not be there. Let me just make it clear. All right. But that's what we need. So I want you to check the description box. You will find that accession ID, and then you can paste it in this side here. Let me just do it again. So we have two entry widgets here. So use this one, the one with enter accession, the second one. So copy the accession ID in the description box and then paste it there. And then after that, you can click on view. So this will send you to the page which has the files. All right. I'll also leave the link to this page in the description box. So you don't necessarily need to search for it. You can use that page, that link, and visit this page. All right. Okay. So this data is Mycobacterium ostrans isolate, a bacteria genome isolate. And so I use this file a lot, this data a lot, because it's the file sizes are very small. All right. So we have paired end reads, and there's also a nanopore data, but we are using the Illumina paired end reads, so that's what we will use. All right. So when we are here, just scroll down to this side. We are going to download the data here. So we have paired end reads, so we will use read 1, read 2 for our genome assembly. So we have them here. Because I'm going to download on the terminal using Nextflow, I will need to get the download links. All right. So I'll first with the read 1, which is this one. So I'll right click and I'll copy the link. And then I'll move to the terminal to download. So I'll move to the terminal. And then when I'm here, I'll create a text file which will contain the download links. So I'll use nano to open a text file. So I'll say nano links.txt. And then I'll open it. You can use any other text, that's fine. Now I am going to paste my link there. This is the first one. I'll move to the next line, and then I'll also go and copy the read 2, which is here. So I'll right click and then I'll copy the link. All right, and I'll move to the text file again. I'll paste it. I'll exit and I'll save changes. I'll still put these links in the description box. Now you can do an ls, you find it there. You can also do a cut just to see the content. That's fine. So now we are going to write a Nextflow script that will download the files for us. And so the Nextflow script will take the file, this file which has the links as input, and then process that for us. Now, before we write the Nextflow script, I want you to take a note of your working directory. So do a pwd and then take note of whatever that has been displayed for us. So we will need this information when we start writing the script. So do that first. All right. Now we are going to write the Nextflow script to download the example data. So we have the links here and we also have the file. So the links to the data are in this file, this text file, which I showed. And so we will open a file and then put in the Nextflow commands and then get the read for us. So let's do this. So let's open our script now. So I'll say nano pipeline.nf and then I'll open it. You can use any text editor. That's also fine. All right, ladies and gentlemen, welcome to Nextflow 101. So we will start by defining some parameters. So I'll start with this one. So I'll say params dot link file equals, and I'll specify the file which has the download links. All right. So I'll bring my quotes and I'll say /home/obna/ Nextflow /links.txt. That is the file. So we have that here. All right, now let's do this. Let's move to the next line. In the next line, we will say params.fascioda equals, we will specify a directory in our working directory. So I'll say /home/kobna/nextflow/fascio, and then I'll bring my double quotes. Now this directory does not exist. All right, that's fine. We are going to create it later on, but we need to define it here. So this directory is where the downloaded files will be stored. All right, so we are going to supply Nextflow with this information also so that the necessary steps are done for us. So for now, these are the information that we need. This is the information that we need. So let's proceed. Now let's scroll down. We are going to now write a command, or let's say commands that will do the download for us. All right, so with Nextflow, if you're going to perform an activity, you need to put it in a process. So because we are going to download files, we will define our task as download. So I'll say process download. And I'll bring a curly brackets to open and then one to end. So in between is where we will place our commands. So take note of that. In between is where we will place our commands. The commands will work together to get us our files. That's what we need. So take note of that. Now let's proceed. So this is what we are going to do next. We will also define a directory. So I'll say publish there. I bring a bracket. I bring a double quotes. I bring a dollar. I bring a curly bracket again, and I'll say params.fascio. Remember we defined that earlier on. Then I'll bring my curly brackets again, the closing one, and I'll bring my double quotes. All right. I'll bring a comma. Don't worry, I'll explain everything. So comma, and then I'll say mode, colon, and I'll say copy. And then I'll bring my brackets again, the closing one. Now this publish that here, this is used to set a directory where output files are stored. Nextflow by default has a working directory. Let me just rephrase it. Nextflow has a default directory that it stores outputs. And so what we are doing is to tell Nextflow that this is the directory that we want, and this is the directory that we want all the outputs to be saved or stored in. That is where the publish dar comes in. All right. Now we define the param.fascio dar here and we specify. So that means that we want the output files to be saved or stored in this directory called fascio in our working directory. So what will happen is that Nextflow will create this directory and then it will store the output files in that directory. So the output files here are the reads. the file that we download, the FASTQ files. Now the mode here, with this publish dar, we have different modes. So the copy here means that we want a copy of the files or of the outputs to be stored in this directory. There are different modes. One of the modes is also move. So move means we will move the file. I made mention that Nextflow has a default directory where it stores output. So the copy here means that the files will be copied from that default directory into our directory of choice, which is FASTQ dar, or FASTQ here. This is the directory of our choice. So Nextflow will make a copy of the output files and place them here for us. All right. You can choose to use move, but that's not recommended until you are done with your analysis. All right. So I will still use copy for now, but maybe if you don't have enough space, then you can choose to use move, but just be careful that always try to use the copy mode. All right. So this is the publish dar. Now let's proceed. Let's scroll down to the next line. So in the next line, we'll type input colon, input colon. And then the next line, space, and we say path. And we say link file. Now the input here is used to define the input, what we are going to place in the process. This is the process, this is the task, but the task requires an input. All right. So the input is going to be our file, the links.txt, but we defined that using param.link file. But in the process, we also need to do additional definition. This is where the input path and link file comes in. So the link file here is a placeholder for the inputs, the incoming information. That's the input. So it's a placeholder. So it's just like, let's say you are in a building. Let's say a security building, and you have IDs. All right. So the IDs are supposed to be given to people who come into the building. All right. So the IDs are a placeholder. And so if someone comes and is assigned that ID, then wherever that ID is made mention of, it refers to that person. So just take it along that line. That is a placeholder for the inputs. So we are defining it here. So in our scripts, in our process, all right, whenever we want to process that input, then we will just make mention of it using this name here. And take note that this name here is different from params.link file in terms of the names. All right, so there are two different entities, even though they are linking the same file. They are two different entities, so don't mix them up. Just follow me, follow what I'm doing here. You can also use a different name. It's up to you. It's just a placeholder, so you can use any name at all. That's what you should know, whether it's file or whatever. But I'll use link file here. Now let's move to the next line. The next line, we will define our outputs. So it's also a colon, space, we say path, and this time we say double quotes star double quotes. All right, so the path and star here indicate that we want all the outputs. So star is everything, so we want everything, all the outputs. That's where the star comes in. Now we want all the outputs, but we also want to make reference to the outputs. We want to display the outputs. So I'll bring this other comma. So I'll say comma, and I'll say emit colon, and I'll say output file. So the emit word here, or command or functional utility, whatever you want to call it. So this emit makes the output file accessible outside this one here, this download process here. All right, so that's why it's the emit. And it also makes it possible for us to make reference to that file or the output file. All right, so in the process, when we want to make reference to this particular file, or let's say this particular output, because we have the output here, then we can use the name. So this is a name, a label for the output file. So yeah, so let me just make it clear. So this is a name that is given to the output, so that whenever we want to make reference to that output, we just need to specify just this name here and it will be fine. So it's also a placeholder for the output. All right, so that's what you need to know. So that is it for the output. Now let's scroll down to the next line, and here we'll define our script. So I'll say script colon, and then the next line I'll bring 3 double quotes, and then another 3 double quotes again. So we have 2 of them, this and this, and in between is where we place in our scripts. I repeat, in between is where we place in our scripts. Okay, so let's do this. So we are now going to issue the command to download the file for us, or the files. We have 2 files, by the way. So the command is going to be this: cat $link file. Remember our link file here, the placeholder. So the link file here is the input. So whatever input we place there, this is it. So the dollar here is like the value, the actual value. So cat $link cat is used to display contents in text files. I bring this pipe. Now say x-axis -i -p2. I'll say wget. I bring a quote, 2 curly brackets, 1 open, 1 closing, and that is it. Now this x-axis command, I have covered it in detail in another tutorial. So if you don't understand this, just watch that tutorial. So basically what this command is doing is to download the files for me. All right. So if let's say you have 10 download links, 20, whatever, all the more reason why I have to use the s-axis. So the s-axis allows us to process multiple files. So let me just put it that way. Simple. It's simple. All right. But still watch that video on x-axis if you have any challenge. All right. The link to that video is in the description box. All right. So just make sure to do. For now, we are using it to download our file. That's all that you need to know. All right. Now let's proceed. So this becomes our final command for downloading the files. All right. Now let's do this. Let's make sure we have everything. We need to also have our curly brackets. A process begins with a curly bracket and ends with a curly bracket, which is the other one here. So make sure to have both of them. Again, you can still download the materials if you want. The link is in the description box. So the scripts are already prepared. You can still download them and then use them or compare with what we are doing now. So let's continue. So this becomes our command for downloading, and we still need to do an additional thing. With the Nextflow version, this version, we also need to specify the workflow. All right, so this will be used to call the processes that you want to run. So we will define that here. So for that, we will say workflow. We also bring a curly bracket opening and then ending, and then in between becomes our working area, sort of. So now with Nextflow, tasks are referred to as process. We've done that here. Now we are performing a task. We are downloading some files, so it's a task. So that's why we have defined our process. But this download requires an input. That is our links.txt file here. All right. So with Nextflow, we supply inputs to processes by using what we call channels. All right. So channels allow us to transfer, allow us to communicate information, allow us to supply information to processes. And so I have some tutorials on channels, so I'll still leave those tutorials in the description box if you are interested to watch that. So basically, this is how I will define channels for now. All right, so we need to create a channel for our input file, and then we supply it to the process download. So the input file, will name it link_ch, and I'll say equals, and I'll say channel.from path, and then I'll specify my link file. So this time around, we are using this one, params.link file. This one here is what we are going to use here. So I'll say params.link file, and then I'll close the brackets. All right. So after defining our channel, we can now call our process download and then supply it with our input, which is now in this channel. Yeah, so I'll call it. So I'll say download, and then I'll do this. I'll bring my brackets and I'll say link underscore ch like this. And then I'll come to the next line. Now, this command can be run, but we also want to know what's happening in terms of the outputs. So I'll indicate another command that will display the output for us. In this case, our files. All right, so I'll say download.out.outputs file.view. Remember, we also specify this here. Okay, so the emit output. So now we can make reference to that file or those files here. And that is what we have done here. Now that is it. So let's just make sure we have everything. Let's check that we have our curly brackets also here. That's fine. So we can now proceed. So that is it basically. All right. So now we can exit and save changes, and then we run Nextflow. So let's exit and save changes. Okay, that is done. So now we can proceed. So that's what we are going to do. Let's do an ls first to check the file. We can also do a cat to display the scripts, and now we can run it. All right, let's just do it again. So now we can run it. So I'll say Nextflow, and I'll say run, and I'll specify the pipeline. So this is it. So let's execute this command. All right, so Nextflow has started this work. So let's wait for everything to get done for us. Okay, the files have been downloaded. So let's take a look at them. So let's do an ls. We are going to find a directory called fastkill. All right, so let's ls into it. We are going to find our files here. All right, so this is how we download it. So that's fine. Now, for the names here, let's go back to this page. I think I have to show you something. So if you download using these links, then you have these names. If you download using these links, then you have these names. So whatever you use, make sure to take note of the names. It's important. But we use this one, so that's fine. You can also try with this one and see what I'm talking about. So don't worry about anything. Now we can proceed. So now we are done with the fastkill files. All right, we have them. So now we are now going to perform the genome assembly. Now, let me also say that if you have your fastkill files, it's important to perform quality control. And this will allow you to identify possible issues so that you can correct them, and then you would have removed low quality reads if you identify those issues, and then you can now use that for downstream analysis. So removing the low quality reads also saves you time when you are analyzing the data. All right, so it's always important to perform the quality control. Okay, but for this tutorial, I'll skip it because the focus of this tutorial is to show you how to use Nextflow to perform genome assembly. So I will later make a tutorial where I show how to also use it for how to use Nextflow for quality control. So that tutorial, I'll show you how to run FastQC. So just stay tuned. So for now, just make sure that we are using this, but we should have done quality control. So quality control, you assess the quality, you perform read trimming and filtering if necessary, and then you are done. All right, so if you have your own data, make sure to do that. All right, let's continue. And let me also say that I have other tutorials on SPADES for genome assembly. So there's one where I showed how to use the Galaxy platform. There's one where I showed how to run SPADES directly on the command line. So if you're interested in those ones too, you can check the discussion box for those videos. So you have a bit of both words. Let me just put it that way. All right, so now we can continue. So let's clear the screen and then continue. All right, so let's do an ls again. Let's do an ls into our fastkill directory to check our files. So we are now going to perform the genome assembly. So we are going to write our Nextflow scripts to perform genome assembly, and this is going to be our input. We have paired and reads. So let's continue. Now, we already have Nextflow installed, and so you should also make sure that you have SPADES installed. Again, instructions for installing Nextflow and SPADES can be found in the description box. So use them to set up your PC. And you should also download the example data. All right. Now let's proceed. So let's do this. We need to take note of some things here. First of all, we need to take note of the path for the files. I have a paired end data. So this is read 1, this is read 2. I need to get a path for both. All right. So I'll just get a path. So to get a path in Linux, you can use the rel paths utility. So I'll say rel path, and then I'll say fastkill / and I specify read 1, for example. So that'll give me the path. And then for read 2, it's still the same approach, but this time the file name changes to 2. So I have them here. Alternatively, you can just use a pwd to get the current directory, and then you add /fastkill, and then you add your / file name here. So that's it. So that's what we need. And we also need to take note of the sample name. We will use it later on. So just take note of that. All right. So for me, I will just take note of this one. I will do everything else in the script. So that's it. Now let's proceed. Now let's open our Nextflow scripts. We are going to continue with this one. So that means that we are going to add on to what we have already done here. So I'll open it. So I'll say nano pipeline.nf, and then I'll open it. All right, so I have that done. Now I'll do this. When I come here, where I initially specify the parameters, I'll add the read 1, read 2, and I'll add something else. Let's start with the read 1. So I'll say params.read1 equals, and then I'll say /home/ so there's a 4 path /1.fastkill.gz. This is for read 1. Params.read2. There's still the same thing here. I'll just do a copy and paste because it's long. I'll just do a copy and paste here. _2.fastkill.gz. All right. Now let's also do this. We are going to add another parameter. So I'll put that also here. So this parameter, I'll say params.spades out, or let's say output. So this directory is where we will place the spades output files. So I'll define it here. So again, it's also /home/ / this /spades output. All right, so this directory, we will use it. So we are also going to use this one. We will use it as we did here, the publish the function here. So don't worry, we are taking it step by step, so we'll get there. So these are the only things I need for now. So now I'm going to add another process for the assembly. So I will scroll down. And then when I scroll down, I'm supposed to scroll down to this side. Let me just go over again. So from here, all right, from the last line of the params, I scroll down to this side. It should come before the workflow. That's how I prefer to do mine. So I'll just do it here. I prefer to bring it before the workflow, this one here. So this is the next process. So I'll define the stuff here. So I'll say process assemble. You can also use assembly, whatever, it's up to you. And then I bring my curly brackets starting and then ending. And then in it, I'll use my publish dar. So I'll say publish dar, and then my brackets, double quotes, $, curly brackets, and I say params.spades outputs. All right, let's just confirm the name. Yeah, so spades outputs like this, and then I bring my curly brackets, I close it, and then I bring my comma, and then the mode. that will be mode colon, and then it becomes copy like this, and then we bring our brackets. All right, so this is how we do it. Okay, so that means this directory will be created, and then the spades outputs will be saved in that directory. Now let's proceed. Let's scroll down. Now we have to define the inputs for this process. All right, let me just do it here. Just make sure you have your curly brackets, yeah. Now we have to define the input. So the inputs here, we will supply two files, read one, read two. So input colon and then space, I'll say path. All right, and I'll say read one. And another one again, path read two. These are the inputs. And then the output colon, and then when I come, I'll say path double quote star double quotes. This is it. All right, so this is it. Now I bring my comma and I'll say emit, and then I'll say outputs. You can put it like this, that's fine. Or you can also be specific or the script. So you can say spirits outputs. That is up to you. So that one, you have to decide the name that you want. All right. So we will do it this way. Now we have to enter the command. So this is where it gets interesting. Let's just go down a bit. So that's a script. You bring your colon, 3 double quotes, 3 double quotes. And then inside, we will specify our spades command. Okay. Now, with spades, if you are going to use it, you also need to specify an output directory. Now, let's take a look at something. We will come back to this script. Let's do an ls into our fastq directory again. We have our fast. Now, with the output, what we want is to supply the sample name minus the .fastq.gz and this _1 here. So _1, _2 are used to differentiate between the for and the reverse. But we want space to create a directory called this, and then everything here is omitted. Everything here is omitted. So we just want to have a single directory with the output. So I'll show you an approach to deal with it. Sometimes it gets confusing for beginners, but I'll show you everything now. Okay, so I'll still take note of these ones, this sample name here. We don't need to paste it. I'll show you everything. So in this script, all right. So ideally for spades, this is how we should run it. Let me just show you the command we are supposed to use. So it's going to be spades.py. And then this is how I'll use it. That's careful. And then I bring my -1. That's for read one. Let me just bring this one here. Read one. -2 $read two. And then -o. All right, it's going to be this one here. Okay, let me just do it. I think this will be better. Say -1. -2. And then I bring my sample, and then -o will be this name here. This is the command that we are supposed to use. All right. Now, if I run this, I mean, on my terminal now, it's going to work. Okay, let's try it. Okay, it's working. That's fine. Let me just cancel it. We don't need it now. And so if that command has been successful, you have all your files, but still, let's do an ls. We are going to find this directory being created. All right, here. Okay, so the command line version worked, but in Nextflow, the command will change slightly. Okay, so this is where you need to pay attention. The command will change slightly for us because we are using Nextflow. All right, so basically, this is what we are going to do. This is what we are going to do. So I'll show you how to write it in Nextflow. So first, let's remove the directory that we created, this one here. We don't need it now, so I'll remove it now. Okay, it's gone. So at the end of the day, what we are going to do is to have a bigger directory, or let me just say one directory, and in it we will have this file, no, this directory also in it, a subdirectory which has the spades output for this sample, right? So that's what we are going to do next. So I thought I'll show you this first before we start now. Okay, so now we are here. So if you are here, you can type your spades, all right, spades.py -- careful, and you say -1, and -1 will indicate our read1, that is $read1, and then -2 is for $read2, all right, and -o is supposed to be the sample name, the output. But at the moment, there's no way. With what we've done, it becomes difficult to get the sample name, all right. Now, under normal circumstance, if you want the sample name, let's just do an ls again to this directory. If you want a sample name, you can process any of them using some text processing utility. You can just do this. Let me show you. I do it a lot. You can just say base name Pascal, and then you specify, let's say, one of them like this, and you can say _1.fascio.gz. all right. So if you run it, you get a sample name. But in Nextflow, you have to do something different, okay, to still get it. So I'll show you that also now. So when we come here, we are going to clean everything. So here, the approach we are going to use is a combination of xargs and then the spades command and then some other stuffs. That's what we are going to do. So here, what we are going to do is to first get the sample name. That's what we will get first. We want to get first, and then we will pass it to the spades command. All right, so to get a sample name, this is the approach I'll use. I'll use the echo command, and then I will do this. I will also bring the read1. That's what I want to use to process. It doesn't matter anyway. If you use read1 or read2, the same principle. So I'll say $. I bring my curly brackets and I'll say read1. And I'll say .simple name. All right. So if I use .simple name, that's what happens. You'll get this up to this one here. But then we also want to remove the _1 as well. So that's what we are going to do. We are going to use the cat command. All right. You can also use the awk. That's also fine. So that's what I'll do. I'll bring this pipe and I'll say cat, and I'll say -d. That's the delimiter. I bring a quotes, underscore, and this. So the delimiter here means that the text will be cut. Wherever there's underscore, it will be cut. So you have a split. So when we split, in this situation, when we split this and this, we are going to have two values, this and then this. But we want the first one. And therefore, when we come here, we will say -f1. We want the first one. All right. And then after cutting it, then we can bring xargs again. So xargs will help us to push the outputs to spades. And then I'll say -i. And then I'll say spades.py -- careful. And then I bring -1 $read1 -2 $read2. And then I'll bring something else. I'll say -o like this. And then I bring a quotes. I bring two curly brackets, one opening, one closing. So this curly bracket is going to be a placeholder for the output of this. The output of this, which is this one here. That is the sax command. Okay. That's how it's going to be. And once we do that, means that everything is complete. All right. So that's why I made mention that when using Nextflow, some things will change slightly. So this is now the final spades command. All right. So we are good to go. Let's just streamline the stuff as well. Okay. Now the x-axis, if you are dealing with multiple samples, then there might have to be some variation in how you use it. But for now, we are dealing with a single sample. So that's why I'm using this approach. All right. But there are other approaches you can also use if you have multiple samples. All right. So most of these commands, it will depend on a number of factors, which include the number of samples and etc. But for now, this one will do fine for us because we have just a single sample. So that's it. So this is it for the process assemble. Okay, that's the task. And then we can continue. We need to also make sure we have our curly brackets there. That's it. All right. So this is the final command for the assembly. All right. So that's it. Now we need to create a channel that will supply this process with the read 1 and then read 2. All right. The publish that will also make sure that we have access to the outputs when everything is done. So let's continue. So now we will come to the workflow. Let me just do this. And then we will continue. Now with the workflow, we still have the command for the downloading of the files, but we don't want to run it again. So you can make it a comment. So in Nextflow, we have single line comments and multi-line. So this time I'm going to comment out all these ones. I don't need them for now. I'm going to comment them. So this will be the command to comment them out. All right. And then I'll rather come down and prepare new ones. All right. Alternatively, you can decide to just remove them completely, but that one is up to you. So you decide, but commenting them is also fine. Now I'm going to run the assemble command by first creating channels to supply that process with the input files. So I'll say read1_ch because channel.from path, and I'll say params.read1. And then read2, let me just add a space here, read2_ch underscore like the ch equals channel.from path, and that will be params.read2. All right. And then when we come down, we can call the assembly process. So I name mine as assemble like this. And then I'll bring my brackets and I'll say read1_ch, comma, read2_ch like this. And then when I come down here, I'll just say assemble.out.spades outputs.view. All right, let's just confirm this name here. You know, we had this emit, so this one here. All right, that's it. Okay, some people may choose to use other ways to display the content, but I prefer to use this one here. So that's it. So once I'm done with this, I need to make sure that everything is correct. I have my curly brackets and everything, and that's fine. So this becomes our final command to perform the genome assembly. Let's just go over again to make sure that the names, everything is correct. So it's fine. Inputs, outputs, yeah, everything is great. Okay, so now we can proceed. So let's exit and save changes. All right, I'll just do an ls again just because I like doing it. And then I'll run the command. So I'll say nextflow run pipeline.nf. All right. So now let's execute this command. Okay, so now Nextflow is running. So this particular activity, when I started it took about 15 minutes for everything to get done for me. So I expect it to be the same for you, or maybe it will be faster depending on the computing power that you have. So let's wait for the Nextflow to complete this task. Okay, so the genome assembly process has been completed. So you will see this log here just to let you know what has happened. All right. Now let's take a look at the output file. So now if you do an ls, you are going to find this directory spades underscore outputs. And if you ls into it, you are going to find this directory which has the output files for that sample. So let's ls also into it so that we see those stuffs there. All right, so we have the files here. All right, so these are the outputs for spades. Now after performing the genome assembly, it's also important to evaluate the genome assemblies. So you can use tools like Quast. You can also use tools like Busco. I've covered some tutorials on them, by the way. I will leave the links to those tutorials in the description box. You can also visualize assemblies by using the graph, assembly graphs. I've covered a tutorial on that. You can also perform polishing. You can also filter the contigs and remove those that are short. You can also perform genome annotation. So I have an entire playlist on post genome assembly. And so check the description box, you will find the link to those videos. And you can also check the playlist here and you can learn more.